Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO. Today, Dr. Jennifer Dwyer will present rapid-acting treatments for pediatric depression and suicidality. Where are we now? BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 183 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the Foundation to find creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research is funded by our BBRF has had such a great impact is because we do not limit our focus to one illness or condition. Since 1987, the Foundation has awarded more than $430 million to fund more than 6,200 research grants around the world across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Jennifer Dwyer. Dr. Dwyer is assistant professor, co-director, and co-founder of the Pediatric Depression Clinic at Yale School of Medicine. She was a 2020 BBRF Young Investigator. Our webinar will begin with Dr. Dwyer's presentation, which will then be followed by question and answers. To submit your questions, please use the questions tab on the control panel on your screen. Feel free to submit your questions at any time during the presentation, and I will ask as many as time allows after the presentation. And now, I'm pleased to introduce Dr. Dwyer. Jenny, the floor is yours.
Excellent. Thank you so much, Jeff, for the introduction and for inviting me to join everybody today. We are talking about a topic that is near and dear to my heart, which is rapid-acting treatments for pediatric depression and suicidality, and really thinking together about where are we now in what is a rapidly emerging field. So what are we trying to accomplish this afternoon? So I hope that by the end of our hour together, you'll be able to describe how novel rapid-acting antidepressant and anti-suicidal medications might work in the brain. I also hope you'll be able to describe what we know about these medicines in pediatric patients compared to adults. And finally, I hope you'll be able to appreciate what the state of the field means for patients and families right now and what we might hope for in a few years. So to accomplish these aims, this is a roadmap of what we're going to be talking about. So I'm going to talk about depression and suicide in adolescents. Then I'm going to talk about interventional psychiatry treatment modalities for depression in adults. Then we'll focus in a little bit on pediatric ketamine, and we'll talk about a single dose randomized controlled trial in adolescent TRD. Then I'm going to spend a fair amount of time really focusing on the gaps in our understanding. What are the key research questions we need to answer moving forward? And I'll wrap up with ways that my group and others have been trying to address those gaps. So let's go. Adolescence is a critical developmental period. It's the period of transition from childhood to adulthood, so conservatively estimated between 12 and 18 years in humans. And there's a unique set of behaviors that are conserved across mammalian species. So from mice and rats to monkeys and humans, you see increased risk-taking, increased novelty-seeking, and spending more time with peers than with parents. But it can also be a serious time with real vulnerabilities. It could be the time of onset for substance use or the emergence of neuropsychiatric disorders. And we really believe that all of these features, the strengths and resiliencies and the vulnerabilities, are mediated by changes in the structural, functional, and neurochemical organization of the brain during this time. The adolescent brain is really a unique developmental substrate. So let's think about major depressive disorder. And I really take a lifespan view of psychiatry in general and major depressive disorder specifically. So we know that depression that begins in adolescence is associated with persistence into adulthood. We also know that pediatric depression is associated with more severe clinical courses compared to later onset MDD. And finally, we also know that early onset depression is associated with increased risks for other problems. So things like poor social and academic functioning, unintended pregnancy, physical illness, and substance abuse. So I really believe that treatment of adolescent depression is prevention for adulthood. How do we make an adolescent MDD diagnosis, and is it any different from an adult diagnosis? This is a figure from a paper that came out in 2020 where we're comparing the criteria for each age group. So on the left, you can see adults and pediatrics on the right. And you might see there's more similarities than differences. So both require 5 of 9 criteria listed here, with at least one of the cardinal criteria as an entry point. A key difference is that pediatric depression allows for not just depressed mood as one of these cardinal entry points, but irritable mood. And so I would encourage practitioners when you see a chronically irritable child to make sure that depression is considered in the differential. So adolescent depression is a significant health problem. So even pre-pandemic, it was estimated that nearly 1 in 5 adolescents will experience MDD before their adulthood. David Brent's work suggests that MDD increases the risk of youth suicide up to 30-fold, and suicide is now the second leading cause of death in this age group. And so already, even before COVID-19, we had a significant public health issue on our hands. But as everyone knows, it's been a very intense and trying couple of years dealing with a global pandemic, and we're just starting to collect the data to really get the numbers to attach to some of the mental health outcomes. So globally, it's been estimated that the prevalence of pediatric depression and anxiety has doubled. If we zoom in geographically and just think about the US, between April and October of 2020, so really one of the first big waves of the pandemic in this country, the proportion of mental health emergency visits increased 24% in kids and 31% in adolescents, with girls at higher risk. If we zoom in again and look at emergency room visits specifically for suspected suicide attempts, CDC data has suggested an increase of over 50% in adolescent girls. Here's some of the data from CDC showing this. So on the x-axis are weeks of the year. So this takes us up through about May. The dotted line is 2019, the dashed line is 2020, and then the solid line is 2021. And you can see a considerable increase in these presentations for suspected suicide attempts. And if you look on the y-axis, just thinking about the sheer numbers, this is thousands of emergency suicidal presentations a week. Here are some sobering CDC facts about suicide, and these were collected before COVID-19. So this was a survey among high schoolers in the US, where 17% reported considering attempting suicide in the last year. 14% reported having a suicide plan in the last year. 8% reported attempting suicide in some way, shape, or form in the last year. And 3% reported making a suicide attempt that required medical attention. The suicide death rate at that time was 13 per 100,000, which is about 5,500 deaths per year. To try to think about those numbers in a more tangible way, I think about, this is a local high school, it's just down the street from my office in New Haven. This is Hamden High. It's a relatively large school, about 1,100 students. And so this is the equivalent of losing 5 Hamden high schools to suicide every year, which I think is completely unacceptable. So I started with saying adolescent depression is a significant health problem, but I think the American Academy of Pediatrics and the American Academy of Child and Adolescent Psychiatry have said it better than I have recently, and have really declared a national emergency in child and adolescent mental health, and a time for us to gather our resources, our expertise, and our advocacy to really make a difference for people. So let's think about treatment. What does adolescent depression treatment consist of? So our first line treatments are really selective serotonin reuptake inhibitors, with fluoxetine or Prozac being the best studied in pediatrics, and combining that with an evidence-based psychotherapy. So an example could be cognitive behavioral therapy, but there are certainly other psychotherapies that have shown efficacy in pediatric populations. But we found from the TADS trial, so this was a major NIMH sponsored trial in the late 90s, that 40% of adolescents with MDD are still depressed even after this first line treatment. So what's next? So second line treatment is to switch to a different SSRI and to add CBT if you have not already. And this data comes from the Tordia trial, which is another large NIMH funded trial. And that trial showed that while a significant proportion do get better, nearly half will remain depressed. And so that makes for about 1 in 5 pediatric patients with depression that will go on to have this treatment-resistant phenotype. And then the question is, how do we treat these patients? And that's where some of the big challenges to evidence-based child psychiatry become readily apparent. So the evidence base in child psychiatry, particularly for pharmacology, is relatively small, and I will say it's been stagnant, really. And of course, lack of evidence has not prevented childhood mental illness, and it certainly has not stopped child psychiatrists from attempting to help their patients. But the result is that off-label prescribing is very common, so we rely on data from adult studies. And our highest risk children are increasingly exposed to interventions with uncertain efficacy and maybe higher harm potential. So one of the examples could be the escalated use of atypical antipsychotics. So this is sort of a depressing picture, but is how I have viewed the pediatric psychopharm research landscape over at least the last decade. And there's reasons for this. These are hard studies to conduct. There are high placebo rates. It can be difficult to find specialized or qualified sites or investigators. In industry, there was a flurry of unsuccessful trials in the late 90s and early 2000s, and many left not only the pediatric mental health space, but just mental health in general. And then the other folks we rely on to fund these kinds of studies are NIMH, and they too had made a shift away during the 2000s from funding clinical trials if there was not a proposed mechanism or target engagement. And the result of that was NIMH had actually not funded a pediatric TRD medication study since Tordia was approved in 2001. And then the question of whether federal funding alone is sufficient to conduct adequately powered trials. But it's important that we find solutions to these challenges. If you prefer data to this sort of depressing graphic, here's a depressing table. This is also from our JCPP review, and it's looking at different augmentation strategies for treatment-resistant depression. You can see on the left-hand side, there's a list of strategies, and really where I want you to focus is on the level of evidence for efficacy. And if you look in the adult column, you see some encouraging letters: A grade evidence, B grade evidence. And when you're looking at the pediatrics column, you know, that's not a good report card. It's D's down the board. And so there really needs to be a concerted effort of specifically testing medications in pediatric populations to make sure that they work and that we fully understand the risks. So I'm going to focus on this bottom panel here, so interventional treatments, and we will move to talking about interventional psychiatry. What is interventional psychiatry? This is a screen grab from Yale New Haven's website for our interventional psychiatry service. Interventional psychiatry was a term I think initially coined in 2014 by Nolan Williams and Mark George, who were down at MUSC at the time. I think maybe there was a little professional jealousy. There was interventional cardiology, interventional radiology, and so we kind of wanted an interventional specialty too. But what does that mean? And I think it really encompasses neuromodulation and rapid acting pharmacotherapies. And so what does that mean? So it includes some old stuff and some new stuff. It includes ECT, which has been around for a long time as a highly effective treatment for depression. It also includes repetitive transcranial magnetic stimulation, deep brain stimulation, and vagus nerve stimulation. So all of these neuromodulation techniques. And then also rapid acting pharmacotherapies that are usually given outside of the traditional oral route. So things like S-ketamine or ketamine infusion therapy, or even brexanolone infusion therapy for postpartum depression. So I'm going to focus on ketamine as sort of the prototypical rapid acting antidepressant. So this is a cartoon of ketamine in the binding pocket of the NMDA receptor. So this is a medication that was traditionally categorized as a dissociative anesthetic. It's a non-competitive NMDA or glutamate receptor antagonist, so it blocks glutamate receptors. And it has a relatively long history in anesthesia, so used at these higher doses all the way up to 4.5 milligrams per kilogram, given quickly over a minute. But over the last 20 years, it's been developed for other purposes in psychiatry. And this has typically been at these lower and slower doses. So 0.5 milligrams per kilogram spread over 40 minutes. So we know a fair amount about ketamine's acute side effects and safety profile from its anesthesia history. So we know that it can cause a transient increase in heart rate and blood pressure, which is dose dependent. It can increase respiratory rate, but at high doses could cause respiratory depression. It can cause nausea, dizziness, or double vision. And it has a range of psychological side effects, including emergent anxiety, dysphoria, dissociation, sensory distortions, or derealization. So why are we excited about it? I just gave you all the negatives about acute ketamine experiences, but the reason that people became excited was this idea of a rapidly acting antidepressant. So this is a figure from the earliest ketamine studies. This is the Berman paper coming out of Yale in the year 2000. And you can see the folks that received ketamine, represented by the black circles, have a rapid reduction in their depression scores relative to the folks in the white circles who received a placebo. And I really want you to focus in on the x-axis here, where we're looking at 24, 48, 72 hours, and understanding that that's a far cry from our traditional antidepressants, where the x-axis extends, past my screen and down the hallway. We're talking about 2, 4, 6, 8 weeks to improvement. So people got really excited about this rapid acting antidepressant effect. The inset is showing that the side effect profile is distinct from the efficacy time course. People also got excited about ketamine around this idea of it potentially being anti-suicidal in adults, even after controlling for its antidepressant effects. So this is a figure from my colleague Sam Wilkinson, from an individual participant level meta-analysis in the American Journal of Psychiatry, where you're looking at clinician scored suicidal ideation. And again, you can see the folks that receive ketamine have a significant reduction compared to the controls in orange. And I think this would be a big deal no matter what age group we're talking about, but I think in child psychiatry, we've lived for a long time with the black box warning and the idea that just because something treats depression, that's not always tied one-to-one with suicidality. And so I think having a medication that is expressly antidepressant and anti-suicidal would be a big help. So ketamine typically comes in a racemic mixture of an S and an R, and the S enantiomer has been developed into a proprietary compound. The S enantiomer was picked for having a higher affinity to NMDA and the potential for less dissociative side effects. But in adults, it was recently FDA approved to be used in conjunction with a standard oral antidepressant in adults with treatment-resistant depression. So how does ketamine work? You know, most of our medications have worked on monoamine systems like serotonin, norepinephrine, and dopamine, and ketamine is different in that it works on the glutamate system. So this simple cartoon tells you everything you need to know. Just kidding, this is probably too complex a cartoon. I'm gonna point out one or two things here, and then in the next slide, I will kind of summarize things in plain English. But the idea here is that on the right, there's a healthy synapse, and there's a couple things to look for. There's an adequate amount of glutamate at the synapse. You see activity of BDNF, which is brain-derived neurotrophic factor. And those together help activate this mTOR pathway at the bottom left, which I think of as like a factory that creates new proteins and allows synaptic plasticity to occur. So that's the right-hand side, that's sort of a healthy synapse. And on the left-hand side, you see more of a stressed, depressed synapse, where you see there's not as much glutamate in the synapse, and BDNF and mTOR are nowhere to be found. And so you might ask, where's ketamine in all of this? Well, it's here in the upper left. And you can see that the hypothesis is that ketamine at these low and slow doses may be preferentially blocking the NMDA receptor on inhibitory interneurons. So instead of just shutting down glutamate across the board, sort of selective in its inhibition, and that actually leads to improved glutamate signaling in the synapse. As promised, plain English. So here the idea is that ketamine enhances glutamatergic signaling and promotes synaptic plasticity. The prefrontal cortex and the hippocampus are key sites of action, and hypothesized mechanisms include this NMDA antagonism on inhibitory interneurons to facilitate intrasynaptic glutamate signaling, and a separate, but I think not mutually exclusive hypothesis about ketamine metabolites, for example, hydroxynorketamine, may directly activate AMPA receptors, another type of glutamate receptor. But overall, the hypotheses coalesce around being pro-glutamatergic. If you're interested in this kind of science, I encourage you to check out the National Neuroscience Curriculum Initiative, which has digestible neuroscience that explains these and other concepts in psychiatry. Okay, so let's get focused back on pediatric pharmacology and ketamine. What information do we have about the system, and should we even be moving forward in this direction? So what do we know? We know that there is significant maturation happening at crucial sites in the brain during this time, particularly in prefrontal cortex. So there's synaptic pruning of glutamatergic synapses. Animal studies suggest that there are relative changes in GABAergic interneurons. So these very targets where we think ketamine is working, maybe sites that are changing during this time, even down to changes in receptor subunit composition. And so again, I feel like we cannot take for granted the idea that just because something works in adults, that it will work or will even work in the same way in developing populations. That really needs to be carefully tested. So we also know that there's preclinical data, so animal studies that suggested that ketamine could reverse depressive phenotypes in adolescent rats. And additionally, we can go back again to this idea that ketamine's been used safely as an acute anesthetic in pediatrics for over 50 years. So with that information, we decided to move forward in asking a pretty straightforward question, which is a single dose of ketamine safe and effective in adolescents with treatment-resistant depression? We called this the KIDS study. It was funded by the folks you see here and recently published in the American Journal. So I'm going to walk through this study. We're going to start with the study subjects. So who did we include in this study? So we were looking for adolescents. We were looking for depressed adolescents. We were looking for significantly depressed adolescents, so a children's depression rating scale score of over 40. We were looking for treatment resistance. And so here we define that as having failed to achieve remission with at least one prior antidepressant trial, although as you'll see in a few slides, most of our participants had tried multiple medications. This was also a ketamine in addition to kind of study. So we had folks stay stable on their psychiatric medications for the month prior to enrollment, and keeping them stable in dose and in agent during the 4-week trial. So who's not in this study? Who did we exclude? So this was an outpatient study. So we excluded people who required inpatient hospitalization, and so that includes folks that had a severe degree of suicidal ideation. So the suicidal ideation in this study was relatively low. We excluded people that had a history of psychotic disorder or manic episode. We excluded people with a history of substance dependence or a positive urine toxicology. Pregnancy was an exclusion, and then anyone that was unable to perform written informed consent. This is a note about controls and blinding. So as I had mentioned in a prior slide, placebo rates are pretty high in depression trial, and particularly pediatric depression trials, but we thought it was important to have a rigorous control. So we used midazolam as an active control. Midazolam is a benzodiazepine that has a similar pharmacokinetic profile to ketamine, so similar timings of coming on and coming off. And the idea is to sort of mimic some aspects of the acute experience of receiving ketamine, but not expecting the antidepressant effects. We put additional measures in place to protect the blind, including having separate safety and efficacy raters. So our efficacy ratings were done at the Child Study Center, and our infusions were done at Yale New Haven Hospital on the hospital research unit. This slide is describing the study design. So this was a randomized midazolam-controlled crossover trial. So a crossover trial means that participants get both the active medication and the placebo in a randomized order, separated by a period here of 2 weeks. So infusion days were on day 0 and day 14. If we zoom into the infusion day, you can see the infusion itself is delivered over 40 minutes by an IV. So this is ketamine at 0.5 milligrams per kilogram, or midazolam at 0.045 milligrams per kilogram. We looked at side effects during the infusion day, so dissociative side effects using a scale called the CADS at 1, 2, and 3 hours. And then we also collected blood to look at ketamine metabolism. As you might imagine, we talked to these participants a lot and did rating scales on the days that you see here. And then in a subset of participants, also did some brain imaging and implicit association tasks the day following the infusion. This is walking through subject recruitment. So we assessed 26 people for eligibility, 19 consented for the trial. Of those, 17 went on to receive the first infusion. So 1 withdrew due to an undisclosed medical condition. 1 person panicked prior to pushing the button for their first infusion and decided to withdraw. Folks were randomized by the investigational drug service. So 11 were randomized to the sequence of midazolam and then ketamine, and 6 were randomized to the sequence of ketamine and then midazolam. On the left, you can see everyone completed their second infusion. And here on the right, 1 person improved after getting their first infusion and withdrew to receive ketamine in the community. This is telling you a little bit about the type of subjects we had in our study, so characteristics of our subjects. So 76% female, an average age of about 15 and a half, although as you can see in the histogram here, we had pretty good representation across the age spectrum. About half were local to Connecticut. Their baseline depression scores were high, so a CDRS of 63 and a MADRS of 33 are high depression scores. The average age of depression onset was 13 years, and the average duration of current episode was 21 months. As I mentioned before, the average number of failed antidepressant trials was about 3 here. So even though the requirement was to have at least 1, on average, people had tried 3 or more. About half of the sample had a history of a suicide attempt, and about 60% had a history of non-suicidal self-injury. And then you might remember that I talked about this being ketamine in addition to, so most people were taking other medications, and this is summarizing what medications people were taking. So about half on an SSRI, another 40% on an SNRI or mirtazapine, around a quarter on antipsychotic augmentation, 1 person on lithium, 2 folks on a mood stabilizer, and 2 people not taking any psychiatric medications. So let's look at the actual data. The primary outcome for this study was the MADRS score 1 day after infusion. And so you can see here that ketamine causes a significant reduction in depression score relative to midazolam. And this is an effect size of about 0.75, which is similar to what's been reported in the adult literature for midazolam-controlled ketamine studies. So the primary outcome was positive. Another key metric we look at is percentage of responders. So being a responder means that you cut your depression score in half or better within the first few days after treatment. And on the right, you can see as a whole, about 70% of the sample showed a response to ketamine. 8 of those people had a response only to ketamine and did not show a response to midazolam, whereas 5 people, shown in the green, had a response to both. And then if you shift your attention to the left side of that graph, again, you see those 5 people that responded to both, and an additional person that responded only to midazolam and not to ketamine. This is another way of viewing that data. I've highlighted, I think, the most relevant comparisons in red. So 8 people responding to ketamine and not midazolam versus 1 responding to midazolam and not ketamine, and 3 people that did not respond to either medication. This data shows the time course. So again, you can see the day 1 effect, but we can also see that on average, depression scores remained improved up to 14 days, which is the longest time point that we assessed in this study. I'm going to show a piece of unpublished data here. I think I had mentioned in the setup slide that a subset of participants did an implicit association task after ketamine or midazolam. So an implicit association test is shown here on the left, and it measures reaction times to assess how quickly a participant can sort various words or pictures that flash on the screen. The sorting speed is thought to reflect how tightly any 2 concepts are associated together. So for example, associating me with happy versus associating me with sad. And the quicker the sorting happens, that's thought to indicate a stronger association between those words. So we had 4 different IATs, 1 with depression words, 1 with words about suicide, 1 about self-harm, and 1 about anxiety. And this is a group of 13 participants. And you can see that there's nice looking separation between ketamine and midazolam on the depression and suicide IATs, whereas we don't see differences seeming to pull out on self-harm or anxiety. So even though the level of explicit suicidality was low in this study, we were encouraged by these preliminary data about implicit cognitions related to suicide. Now I'm going to talk about some of the side effects we saw in that trial. I think the alternate title for this slide is that midazolam is not a great control for ketamine. So you can see that there's a significant difference in dissociative symptoms in ketamine compared to midazolam-treated subjects. But you can also see that these are self-limited and are back down to baseline by 2 hours, and there were no sort of prolonged dissociative symptoms at 24 hours. This slide is singling out additional items or individual items on the CADS and comparing ketamine to midazolam just to get kind of a sense of some of the experiential components. And so I've highlighted shared symptoms in the same color. So you can see with both ketamine and midazolam, a high percentage of people report feeling like they've spaced out. There's also some overlap in feeling disconnected from your body, but then there are some symptoms that just seem very specific to ketamine. So at the top, you can see 88% of participants reported feeling like they were in a dream. And then lastly, you'll remember that ketamine can cause changes in hemodynamic parameters. And so this is looking at systolic and diastolic blood pressure as well as heart rate. And showing that ketamine does indeed increase blood pressure and heart rate in the adolescent population. So 4 participants had blood pressures meeting criteria for adolescent stage 2 hypertension during the infusion itself, but none persisted past the end of the infusion and none exceeded 150/95. So what are the single dose conclusions? So overall, ketamine was well tolerated in this small sample. And ketamine significantly improved depressive symptoms at 1 day post-infusion as measured via the MADRS in adolescents with TRD. We didn't have any information here about explicit suicidal thinking since significant suicidality was an exclusion in this study, but we do have some interesting hints from the implicit association data. So this preliminary data suggests that ketamine may be safe and effective in the short term, but like adults, as we followed some of these participants informally after the study was over, the effects of a single dose really appear ephemeral. And so I think the real conclusion here is that more research is needed as the gaps in our knowledge are considerable. And so now I'll shift for this last part of the talk and really focus on some of the gaps that keep me up at night that I think are really important for us to start trying to answer. So let's look at these gaps. So the first gap that I think about is about extending ketamine's antidepressant efficacy and this idea of repeat dosing. So our clinical trial was a single dose, and at this point, most dosing paradigms in adults use a repeat dosing paradigm. So this is data from a paper in 2016 that's looking at a repeat dosing paradigm. Again, ketamine's in the green and placebo is in the orange. And you can see a reduction after the first infusion and an improvement beyond that as subsequent infusions progress. Another study showed that twice-weekly dosing over 3 weeks yielded remission thereafter that lasted for an average of 3 weeks. But when you dig in a little deeper, there's a big range of durability. So some people relapsing in 6 days and some people lasting greater than 3 months. And the individual factors that predict duration are not well understood. So we have some experience using repeat dosing with adolescents. So we published a case report in 2017 using a repeat infusion paradigm. So this was a 16-year-old boy that had a history of ADHD, depression, and Crohn's disease. He had 3 prior serious suicide attempts. He had tried multiple pharmacologic treatments, psychotherapies, and across a range of different levels of care, and was considering ECT at the time, but instead opted for a trial of ketamine. This is an adaptation of a figure from that paper. So in the purple, it's showing the MADRS, again, a measure of depressive symptoms. And in the red is showing the SSI, which is the scale for suicidal ideation. The x-axis is showing the days of treatment, and the arrows indicate individual infusions. And again, you can see a nice reduction after the first infusion and then further improvements with multiple doses during this acute series. People often ask about this blip in the middle where scores go up around day 30, and our ratings that day corresponded to an insurance denial for the discharge plan. And I think everyone's frustration was quite high. And I think that's an important point, that ketamine can be very helpful for depression in some cases, but it does not immunize you from feeling disappointed when disappointing things happen. But you can see that these scores went back down without an intervening infusion, and overall this patient did well. So why aren't we just jumping into repeat dosing? What are some considerations about repeated dosing? And so these considerations come from really two main sources. So one are from studies of heavy ketamine recreational use or abuse. And we see that when people have used lots of ketamine, there are concerns about neurocognitive effects, so neurocognitive damage. Also bladder damage or cystitis. So ketamine has a metabolite that's very irritating to the urothelium. And then of course, just the question about abuse potential in general. So that's one set of literature that makes us think about repeat dosing. And then the second, which is more specific to developing populations, come from animal studies. In animal studies using chronic dosing, usually at higher doses, but chronic dosing in young animals shows damage to areas like hippocampus and prefrontal cortex, and significant things like cell death and white matter changes. Animal studies also have used ketamine and related compounds to try to model development of psychosis and schizophrenia. We hypothesize that this is by blocking NMDA receptors at the synapse at these higher doses, but all of these things give us pause because the toxicity threshold is really not well defined. So this is a general figure that's just looking at how we think about medications and how as the dose increases, we worry about toxicity. And so in the ketamine field, we're anchored by some facts, right? So on the left at lower doses, the pediatric anesthesia literature suggests, that we should feel relatively comfortable about single treatments at reasonable doses. And then at the other end of the spectrum, the abuse literature and these animal studies suggest harm with chronic exposures and higher doses, and that vulnerability may be heightened at younger ages. But what we really want to know is where that threshold is for a population and for an individual. And so that leaves questions about both the dose you're receiving in a single treatment and the number of doses over time. So that leads to the second gap, which is also related to the idea of repeat dosing, which is what to do after an acute ketamine treatment series. So you've received 4 or 6 or 8 doses over a span of weeks. What next? So one option is the idea of maintenance ketamine infusions. This is in many ways following sort of an ECT model of giving a burst of treatments at the beginning and then spacing them out in sort of maintenance intervals. But even in adult psychiatry, the question of how many treatments and what intervals, these are details that are still being studied and worked out. A second idea, which I find appealing, is the idea of whether we can harness the period of wellness in ketamine responders using more traditional psychiatric treatment approaches. So this is another busy cartoon to remind you about the idea of ketamine enhancing synaptic plasticity or synaptic plasticity potential. And so one question is whether we can use that period of enhanced synaptic plasticity to allow for something like psychotherapy to be more impactful. And there's now a number of studies trying to pair intensive psychotherapies with ketamine or just after ketamine treatment to really test that hypothesis. So that's gap number 2. Gap number 3 Do antidepressant effects hold in larger pediatric samples? So this cartoon says, after 19 additional trials, of course, the results were shown to be anomalous. It says the tortoise and the hare is actually a fable about small sample sizes, which I think is a clever cartoon that speaks to a serious issue, which is, the history in our field of getting excited about small studies with positive results, and then seeing those not pan out in larger studies, or it taking a long time for the larger studies to be done and having lots of people sort of treated before the data is in. And so I think it's critical that multiple research groups are starting to do larger scale clinical trials in this space. And then a related question about efficacy is, what about suicidality? And so, as we talked about in the first trial, suicidality was not a big component. And I think it's really important for us to be specifically looking at ketamine's effects on suicidal thinking in adolescent populations. Okay, the fourth and last gap that I will talk about is ketamine for whom? And there's a lot of different ways that we could think about this, right? So which disorders? So we know there's good data in MDD. People are also doing studies in OCD and anxiety disorders and PTSD and others. Is there a particular disorder that is the best fit for ketamine treatment? So that's one way of thinking about it. Another way could be thinking about what kind of clinical features or predisposing factors might be make ketamine a good fit for one person versus another. And again, the adult literature shows us things like anxious depression seem to be a good fit, having a strong component of anhedonia, and then even recent data suggesting that a history of childhood trauma portended a good response to ketamine. So clinical features is another kind of thing we could think about. And then the last way of thinking about this, which is particularly interesting to me, is what biological features? Is there something about the way someone's brain is organized that could help us determine whether someone would respond better to ketamine or an SSRI or a psychotherapy? And this is really, of course, the goal of a personalized medicine approach. I think patients and doctors have all experienced the frustration of our current way of doing business here, which is trying something, and if it doesn't work, you try something different, and if that doesn't work, you try something again, which can result in a lot of lost time. And so if we were able to really predict beforehand, it would be a huge, huge gain for the field. So summarizing the gaps, how to extend ketamine's antidepressant efficacy, particularly as the risks of repeated dosing are not fully known. How to optimally extend efficacy after an acute series, particularly using more traditional psychiatric approaches. Does ketamine genuinely have antidepressant and anti-suicidal effects in pediatric populations? And personalized medicine approaches. Who are the best candidates for ketamine, and can we predict treatment responses ahead of time? So I will spend the next 5 minutes or so talking about ways that my group and others are trying to address some of these gaps. So I'm going to tell you about 2 repeat dosing clinical trials that we've been working on. So the first we call the SAD Kid study, which stands for Severe Adolescent Depression Ketamine Intermediate Duration Study. You can probably tell that we spent a really long time thinking about that acronym. And this was funded by the American Academy of Child and Adolescent Psychiatry and the Klingenstein Third Generation Foundation. And then a related study, this is a bigger study that was funded by NIMH that we've been calling the READS study, so Reducing Adolescent Suicidality, Safety, Efficacy, and Connectome Phenotypes of IV Ketamine. And I talk about these 2 together because they have very similar designs to them. So I'm going to tell you about those designs. They're 2 phase studies where the first phase, which occurs over a number of weeks, is a parallel designed randomized controlled trial. So people are enrolled and they're randomized to get repeat ketamine infusions or repeat midazolam infusions. But then at the end of that period of time, we have a scheduled breaking of the blind. So folks that were assigned to ketamine move straight through to an open phase where they receive standard of care depression treatment, so medication management and CBT. There are weekly mood assessments and monthly cognitive mood batteries. And then for the folks that were assigned to midazolam, if they remain depressed or have significant suicidal thinking, so did not have a response to midazolam, we offer them the option to cross over to receive open ketamine and then to roll forward into the open treatment as everyone else does. And I really like this design because I think it balances the rigor of a parallel design clinical trial with what's really an ethical obligation to offer active treatment to people that are suffering. So there are some key differences between these two studies. So SAD Kids, it's really focusing on pediatric treatment-resistant depression. The dosing paradigm is 6 infusions followed by a limited number of symptom-triggered maintenance infusions, and the primary outcome there is depression using the Children's Depression Rating Scale. And then the READ study is focusing on people that have both pediatric TRD and a recent suicide event. And that dosing paradigm is 4 infusions, and the primary outcome there is suicidal thinking using the ideation subscale of the Columbia. So the last thing on this busy slide, you might see this little brain that just appeared all the way on the left-hand side. And so I'm excited to tell you a little bit about how we've integrated brain-based predictors work into these clinical trials. So this is through a collaboration with my colleague and mentor, Todd Constable here at Yale. And he has developed a very cool technique called connectome-based predictive modeling. So what does that mean? So this is basically using a combination of task-based and resting state fMRI to try to tweak or stress the brain across neurocognitive domains. And the way he always describes it to me is like, if you're trying to understand how someone's heart is functioning, if you just get an X-ray, you just get a static picture, you might not see the whole idea. And so that's why you get people on a treadmill and get a stress test and really look at how the heart performs when it's, doing its duties. And so the analogy here is really trying to give a stress test to the brain. And so we're tweaking it across these RDoC domains and use this to generate a highly individual connectome fingerprint, so like brain snapshot, and then trying to use that to associate it with clinical dimensions of interest. And we've gotten the scan time to under an hour, which I think is good. And he's used this approach to predict things like inattention in an ADHD sample, autism symptom scores in mixed samples of ADHD and autism. And then colleagues here at Yale, for example, Sarah Yip's lab, has started to use this technique to try to predict treatment response. So she was able to show prediction of treatment response in cocaine use disorder. And so that's very encouraging, the idea of using pre-treatment connectome signatures to try to predict post-treatment responses. And so we hope that this is one step towards personalized medicine, or at least laying the foundation for personalized medicine. And so I'll end on my last few minutes kind of talking about the future. I know I showed you that really depressing picture of a wasteland, and this is the world that we're living in, but I'm very encouraged about the future. And seeing things like NIMH making a big investment in trying to understand pediatric depression and suicide and really focusing on studying interventions. And so this is a list of studies, mine included. So I told you about the READ study, but I'm very encouraged that there's more groups out there that are going to be doing these clinical trials. So a trial for ketamine at Cleveland Clinic, and then Madhukar Trivedi's group at UT Southwestern. And I know I've focused here on ketamine, but even rTMS, so my colleague Paul Corkin at the Mayo Clinic is starting a TMS study for pediatric suicidality. So I'm actually quite encouraged about the direction that this field is taking. So when I think, what's the current status? I think cautious optimism. I hope that in a few years, I guess 4 years from now is 2026, I expect that there will be several solid data sources reporting outcomes in this space. So there are several studies at academic centers that are starting right now. I created this QR code, which takes you right to a search page on clinicaltrials.gov. And that lists the ketamine pediatric trials that are going on. And so I really encourage people, if they're thinking about treatment in this space, to see if a clinical trial is available to them. There's also a pediatric study of S-ketamine intranasal spray that's slated to finish their recruitment soon. And I hope that these studies together will give us information about the efficacy and safety of conservative repeat dosing paradigms, so ranging from 4 to 8 treatments, and will give us some short and intermediate term outcomes. Okay, but then you might say, all right, well, that's 2026. What about right now? And I think it's kind of a tricky time in this space. I remain cautiously optimistic, but I think the unknown risk profile of repeat exposure makes it hard to have risk-benefit conversations. And the off-label availability of ketamine in the community is really far outpacing the evidence base. So in a way, I feel like it's a little like Wild West out there. And so the things that I think about for the here and now are really making sure that you confirm treatment resistance. So carefully reviewing past treatment trials and making sure that people have had a good shot at first and second line medications and high quality psychotherapy. I think it also is very important to have informed consent about the risks, benefits, and the state of understanding about the field. Where are we now? Where might we be in 5 years? Direct supervision by physicians with adequate training is critical, so no take-home ketamine. I think that's a bad idea. I also feel that people should stick with the most well-studied dose and not exceeding that 0.5 milligrams per kilogram over 40 minutes. And then this last piece I think is really important, regardless of whether it's ketamine or rTMS or ECT, having a robust follow-up plan. And that should really include a biopsychosocial framework. So not just what medicine is going to be coming next, but how do we find a good psychotherapy match? How do we optimize psychosocial stressors? You know, many of our kids have been out of school for a period of time when they come to one of our treatment trials, and a big component is trying to help overcome that inertia of trying to get back to school and relationships and really getting back on developmental lines. So I will stop there, just thanking some people. So first and foremost, thanking our patients and their families. A big thank you to the people that are funding the work, including the Brain and Behavior Foundation for inviting me today to talk with you. This is a picture of the lab from the summer. It takes a lot of hardworking people to get this kind of work done. I have an amazing team. And then if you're interested in treatment studies, I'm putting the QR code back up. Again, that takes you to clinicaltrials.gov. And folks that are local to Connecticut are welcome to email us at pddepression@yale.edu. Thank you so much for joining me.
Jenny, I want to thank you for joining us. What a great presentation. And thank you for all the work that you have been doing on this important topic and the work that you're going to continue to do, which will have an impact on so many lives. You covered so much. I have to tell you, the questions that came in, you covered those questions in the time allotted. So I'm not going to ask those questions because you've already answered so many of the questions that we received in your extraordinary presentation. So thank you. And I am going to ask you to come back and give us an update hopefully even before 2026 with what you're finding out, because to me, the work that you're doing is so important and so exciting. So thank you so much.
Well, thank you so much, Jeff. I really appreciate your support of the work and your interest, and I look forward to staying in touch.
Great. I also want to thank everybody for joining us today. I ask that you please consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate grants. This means that when you donate $1 for research, that dollar goes directly to the scientist. To make a donation, please visit our website, bbrfoundation.org, or call us at 1-800-829-8289. The webinar has been recorded. If you've missed any portion or would like to share it with friends and family, please visit the events and webinar page on our website. And I hope you'll join us again next month on April 12th at 2 p.m. Eastern Time, when Dr. Denise Kai, Assistant Professor in the Department of Neuroscience and the Friedman Brain Institute at the Icahn School of Medicine at Mount Sinai, will present How Does a Fearful Experience Alter Our Past Memories? Thank you. And remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Thank you.